The virus that causes monkeypox was first discovered in 1958 in a group of monkeys that were used for research in a laboratory. And that's how the virus got their name. Later, in 1970, the virus was first detected in human beings, in the Democratic Republic of Congo. Since then, cases of monkeypox have been reported in other Central and Western African countries. However, the virus is believed to spread to human beings, by rodents, not by the monkeys. Monkeypox virus is an enveloped, double-stranded DNA virus. It belongs to the orthopoxvirus, genus of the Poxviridae family. There are two distinct genetic clades of the monkeypox virus: the Central African clade, and the West African clade. The Central African clade has higher fatality rate than the West African clade. The monkeypox virus can also be found in squirrels, Gambian pouched rats, dormice, non-human primates and other species. There is no clear understanding on the natural history of monkeypox virus, and yet to identify the exact reservoirs, sources of infection and transmission patterns. Until 2002, the virus spread has been confined to African countries. In 2003, the first case of monkeypox, outside Africa, was identified in United States of America. The virus infection in the USA was associated with prairie dogs. These pets were housed in the same enclosure, where there were Gambian pouched rats and dormice, that were imported from Ghana. In the later years, the virus has been spread to countries like Israel, United Kingdom, and Singapore. By the end of May 2022, several cases were reported in other countries too. Monkeypox is a zoonotic disease, which means, the disease spreads from an infected animal to human, by direct contact with blood, body fluids, and cutaneous lesions. People who eat inadequately cooked meat, and other products of infected animals, may also get infected. People living near forest areas, may have indirect, or low-level exposure to infected animals. The virus can also spread by the human-to-human interaction. The human-to-human transmission can occur from close contact with respiratory droplets, skin lesions of an infected person, or recently contaminated objects. Transmission via respiratory droplets usually requires prolonged face-to-face contact. And that puts healthcare workers and members of family at greater risk. Transmission can also occur via the placenta, from an infected mother to baby, or by close contact after birth. The incubation period of monkeypox may range from 5 to 21 days. The entry of virus will be characterized by fever, intense headache, lymphadenopathy, back pain, myalgia, and intense asthenia. The early symptoms of monkeypox are similar like chickenpox and smallpox. However, lymphadenopathy is a distinctive feature of monkeypox. Later, the skin eruption begins, within 1 to 3 days of appearance of fever. The rashes may appear all over the body, but more on the face, hands, palms, and feet. The rashes evolve from macules to papules. Some filled with clear fluid, and some with pus. Then turn into crusts, and then fall off from the body. The symptoms last from 2 to 4 weeks. The fatality cases of monkeypox are rare in the world. Although, the rashes are similar to the chickenpox and bacterial skin infections, the monkeypox can be differentiated from chickenpox by the swelling of lymph nodes. When monkeypox is suspected, the health workers shall have to collect the samples, and send to appropriate laboratory tests. PCR, or polymerase chain reaction test, is recommended to diagnose monkeypox virus. Sample must be collected from the surface of skin lesions, or lesion fluid, and dry crusts. PCR for blood samples is not recommended, because of the shorter duration of presence of virus in the blood. Swab samples can be collected with dry swab, and addition of viral transport media is not recommended. Because of the serological cross-reactions, the antigen and antibody tests are not recommended for monkeypox detection. There is also a chance of getting false positive results, if the person has been vaccinated for smallpox previously. The interpretation of test results may require the patient's age, date of onset of fever, date of onset of rashes, and present health condition. Currently, there is no specific treatment available for monkeypox. Care should be taken to reduce complications and further development of symptoms. Tecovirimat, also known as ST-246, is an antiviral drug used for treating smallpox, was licensed for monkeypox, based on human and animal trials. Currently, this drug is not widely available. The primary mode of transmission of disease is through close contact with infected person. Therefore, healthcare workers and members of family are at high risk of infection. Extensive precautions must be taken, while collecting samples and caring the patients. The staff, who are involved in handling patients infected with monkeypox, must be properly trained, and previously vaccinated against smallpox. As there is a risk of zoonotic transmission, direct contact of wild animals, dead or alive, must be avoided. Additionally, animal food must be thoroughly cooked. Importation of animals must be regulated, and infected animals must be timely isolated. It is demonstrated that vaccines against smallpox are 85% effective in preventing monkeypox. Therefore, prior vaccination against smallpox may prevent monkeypox or reduces its severity. Smallpox vaccines are not available to the general public, except for few healthcare workers. Currently, a new vaccine for smallpox was developed and licensed. The vaccine contains vaccinia virus, which belongs to poxvirus family.